Professor Paul Barrow, could you tell us a little bit about your story in poultry health research, the institutes and universities you work, and a little bit about your career in microbiology for our audience to learn about you?
Okay.
So please.
Yeah, thanks very much, Raphael. It's also good to be talking to you and also to your colleagues through this online system. So I was thinking the other day, that I've been doing research for 46 years, which is a long time, I guess, is longer than some people's lives. And so I graduated. In fact, I'm not a veterinarian. I'm a scientist. In fact, I did a microbiology degree, followed on medical and veterinary microbiology. I did a PhD on pig enteritis, working at the UK Institute and at the University of Reading, and then I did postdoc working on bovine tuberculosis, and I spent 1 year at Unilever Research working on coliform mastitis, and then I was very lucky enough in 1981 to join what was then the Houghton Poultry Research Station. So it was the best poultry institute in the UK, and it was also working with H. William Smith, and William Smith was probably globally, I guess, probably the most important veterinary microbiologist in the world, I think. So he did huge amounts of really good work understanding the pathogenesis of E. coli enteritis, developing vaccines for cattle and poultry, and also, for example, he developed the 9R vaccine. He discovered that many virulence determinants are plasmid mediated, and then in the early 1980s, he also was the first person to resurrect the idea of using bacteriophages. So he was a brilliant scientist. He was a very difficult man, actually, to work with. As some of these brilliant guys are, but I learned a lot. And so I was there working with him for 6 years. Then he died, in fact, and then I carried on working at the institute until 1991. And then at that time, the UK government was going through a lot of reorganization of its agricultural science. So they closed our institute down and moved us down to the Institute for Animal Health at Compton, which is south of Oxford in the UK. And I was there as head of the Salmonella and the Zoonosis Research Group until 2006. And at that time, and we did some really nice work at that time and during that period. At that time also, the Ministry of Agriculture, DEFRA as it's called, Department of Environment, Food and Rural Affairs, were gradually reducing their contribution to science. And so I was reaching the difficult position where I had 2 technicians working for me. One had been with me 20 years and one 15 years, and I was going to have to release one. I wasn't happy about this. So, and then we found that the University of Nottingham was starting a new vet school. So I contacted them and I contacted the dean and said, is there any interest you have in research? And he said yes. So, in fact, I was lucky enough to take the whole research group with me, myself, postdocs and technicians. We all moved up to Nottingham. And in 2006, I moved to the University of Nottingham with my whole group, and I was research director there for 10 years and director of global engagement, which is the international aspect, for another 2 years. And then I stepped down to concentrate on my own research. So I guess over the years, I've been working on salmonella in poultry for 39 years, I think. And I was trying to think the other day of the contributions we've made. And I guess in the early years, when I was working with Smith, we were particularly looking at the effect of growth-promoting antibiotics on salmonella excretion. And several of them, exacerbate, make the situation worse and make salmonella excretion heavier. And so this led or contributed to withdrawing growth promoters in the EU, growth promoting antibiotics.
Around which year or which decade, do you remember? You were already working with that kind of research?
We were working 1981 to about 1990, I think. And then with that, our work was one factor, but in fact, one of the major factors also was that the use of some antibiotics, particularly virginiamycin and avoparcin, they contribute to increased levels of resistance to glycopeptides, particularly vancomycin in enterococci.
Okay.
And a friend of mine in London showed that the resistance in the enterococcus can be transmitted to Staph aureus on the surface of a guinea pig. We also showed that pig and chicken enterococci, vancomycin-resistant enterococci, can colonize the human gut. And so we did that work. And so the medical and veterinary authorities in Europe were very worried about vancomycin-resistant Staph aureus developing. So they decided in 2006 to ban growth-promoting antibiotics from poultry and pigs. It hasn't happened everywhere. So in the United States, there was a big argument. We had argument with them. They said the use in veterinary is not a big problem. It's overuse of vancomycin in human medicine. And we thought, okay, well, that contributes as well. But should we just wait until we get a vancomycin-resistant Staph aureus and then do something? We would rather be proactive and act ahead of the problem arising. So, so our work, I guess, contributed to that. We also, I also showed that the vaccine which Smith developed in the 1950s, the 9R vaccine, which is used extensively against Salmonella gallinarum, we showed that it was also effective against Salmonella enteritidis. And so that was used quite extensively, I think, against enteritidis. Because there was such a big problem, food poisoning caused by Salmonella enteritidis arising from poultry. True. And some of the other things that we've done, I guess we developed an ELISA for use with for serological testing for poultry for Typhimurium, enteritidis, gallinarum, and pyllorum. And we did some of this work, particularly with gallinarum and pyllorum, with Professor Berchieri, who was a very old colleague of mine at
mine as well, yeah.
Yeah. And we also sequenced the gallinarum and enteritidis genomes, which produced some really interesting results. But we've also done extensive collaboration when I was at Houghton and Compton. And one of my greatest colleagues was a geneticist called Nat Bumstead, who unfortunately has now died.
Oh, okay. And
we did a lot of work on the genetics of resistance to Salmonella. And we showed that resistance was a Mendelian characteristic. It was inherited, it was dominant. And that was resistance to systemic salmonellosis. But we also showed that resistance to intestinal colonization by Salmonella was also inherited in a Mendelian fashion. And one of the very interesting things was that we found the same lines of chickens were resistant to colonization by Salmonella and also to Campylobacter. So it's suggesting that certainly certain aspects of colonization by these two organisms in chickens is the same. And that was really quite an interesting discovery.
And did you also work with? Did you also study resistance against Salmonella gallinarum in white and brown lines of chickens?
So we did this with Professor Berchieri, and we found, as far as I recall, that white egg layers are generally more resistant to gallinarum than brown egg layers. And I mean, I'm not sure the significance of this in terms of epidemiology in Brazil, but it may well be that in the case of brown egg layers, that gallinarum is transmitted more frequently horizontally, and in white egg layers it might be transmitted more frequently vertically. Interestingly, also, we've had a long interest in gallinarum and pyllorum, and many years ago we managed to very easily, I guess, establish an in vivo infection model with Salmonella pyllorum in a brown egg layers, and so we showed that we could get persistent infection until the birds became sexually mature, and then the organism was transmitted vertically, and started off with infection of the progeny again. We tried very hard to show vertical transmission with gallinarum. We tried different strains of bacterium, different lines of chickens, different doses, and we found generally that birds either became diseased or they weren't infected at all. But with one particular line of chickens, a resistant line, we were able to show persistent infection for up to, I think, something like 12 weeks, 12, 14 weeks.
Wow.
So we think it's possible that vertical transmission happens more frequently with Gallinarum in more resistant birds, but we haven't been able to continue that work, unfortunately. So I remember discussing this with Angelo Berchieri, that we speculated that maybe in brown egg layers horizontal transmission was more important, and in white egg layers maybe vertical transmission was more important.
Yeah, this is some discussion that happens a lot about Salmonella, especially about Gallinarum, not Pullarum. But if you read older papers, they talk about Salmonella Gallinarum being transmitted both vertically and horizontally.
Yeah.
And the more recent papers talk only about horizontal transmission with Salmonella Gallinarum. So there's something that there seems to be some scientific data seems to be they're not matching, something that we discuss a lot in the field. So do you believe this Gallinarum is really only horizontally transmitted, or depends on the chicken line you work with? They may be vertically transmitted, because it influences a lot in the results we see in the fields, in the epidemiology of this bacteria, if it's vertically transmitted.
I remember one of the other people I knew very well was Professor Frank Jordan in the UK, and he, when he retired, he gave me all his old papers on Pullarum and Gallinarum, and I remember going through them, and so he did some experiments where he infected adult hens and then showed transmission to the next generation. But it was very difficult to see whether it was true vertical transmission. I did wonder whether there was an opportunity for contamination and pseudo vertical transmission, but really horizontal transmission.
Okay.
So it hasn't really been proved, but the evidence is epidemiological, and there hasn't been
really strong, right?
Yeah, there hasn't really been any sound and very strong experimental evidence showing that Gallinarum has been transmitted vertically. But there's a lot of circumstantial evidence, and the evidence in white egg layers, for example, and that in a more resistant line of chicken you can get persistent infections, suggests that it does happen, but maybe much less frequently than it does with Pullarum.
Yeah, okay, yeah. Pullarum is very easily transmitted by the vertical route, right? So one thing you said in the beginning of the interview, you started work with Salmonella around 1990, 1991, something like that. So what do you see over the years? What do you think has changed about the view of this pathogen? And I know that in the 90s you probably lived that, because you started working Salmonella at that decade. So you saw the pandemic of Salmonella enteritidis, and what do you think has changed in the current moment in relation to the dissemination of the Salmonella in the fields? Is Salmonella enteritidis still important for poultry? Do you believe there are other servers coming out now that may be very relevant for our field?
Historically, the serotype of Salmonella, which is predominant in poultry and which causes food poisoning, it does change over the years. So before I started working, Salmonella agona was a big problem in poultry. When I started working just before, I guess, in the 1970s, Salmonella hadar got into the poultry industry, into the turkey industry, and from there it transmitted to the broiler industry, and so that was a big problem. Typhoemerum has always been a problem, and then in the late 1980s, enteritidis became a problem, and this was really interesting because it involved several different phage types, and in different parts of the world also. So we had Salmonella enteritidis phage type 4, 6, 8, 13A, and in different parts of the world, different phage types were important. And it seems rather odd that at the same time, simultaneously in different countries, enteritidis became a huge problem. Nobody has really understood this satisfactorily. There was one school of thought put forward by German colleagues that as palorum and gallinarum were eliminated, because serologically and taxonomically they're very closely related to enteritis.
Yeah.
Maybe this allowed a vacant niche for enteritis to colonize. I don't believe that at all, because certainly in the European countries, palorum and gallinarum was eliminated in the 70s, 15, 20 years before
enteritis.
Yeah.
Okay.
So, in European countries, I guess we've more or less controlled enteritis through legislation, improved biosecurity, eradication schemes, vaccination, etc. It still is a problem in some Eastern European countries, and but now we have other serotypes such as Salmonella infantis, which is a very good colonizer. It's not terribly virulent for chickens, which is also appearing in southeastern European countries, and then it's spreading to other European countries also. The situation in Australia is really interesting because they were able to keep Salmonella enteritis out of their poultry industry, and they did have a serotype, Salmonella sophia, which seemed to be unique to Australia, but it was in the poultry industry, but never caused any food poisoning. So that serotype would be very interesting
to study a little bit. Yeah, we've seen we've seen this behavior in the poultry industry around the world. It seems to be changing, and if you look at the CDC reports or the EFSA, the European Food Safety Authority reports about Salmonella, we see there's some mismatch of the main serovars found now in humans and in animal source, especially in poultry and eggs, are not matching anymore. So the Salmonella typhimurium and enteritis is still highly relevant in humans, but in the fields in poultry industry, it seems to be that other serovars are coming out. In Brazil, for example, we have a lot of Salmonella Heidelberg. In other countries, that Heidelberg is a huge problem as well. So it seems to be changing. So a lot of people ask about this behavior of Salmonella. Why one just vanishes and the other one takes place? So we were talking about this relation. Maybe vaccines interfere. Some people talk about this interference when you use some vaccine controls like gallinarum. Another vaccine may control Salmonella enteritis. But do you believe there's a niche that you open a path for another serovar to come out?
It's very difficult to understand this, isn't it? And the epidemiology is very complex because between the poultry farm and the supermarket, there's also the abattoir in between, and people don't really understand what happens at the abattoir in terms of whether you have serotypes which are predominant as well. So I can't say what the reason for the discrepancy is between the situation in poultry and in food poisoning, and it's really an area that needs to be studied, because I think it does indicate that we don't understand things very fully, and I think if we did understand them better, we might be able to control them a little bit better as well. In fact, I remember that there is a problem with Heidelberg in Brazil, and it's quite interesting because it's serologically again very similar to Salmonella typhimurium. It's a group B Salmonella, and it would be interesting to look to see whether the available typhimurium vaccines have any effect against Heidelberg. I don't know whether people have tested that at all, but it might be interesting to look at.
Yeah, that's something that may be better studied, definitely. So in UK, I think most of our experience with Salmonella control comes from Europe and UK. We have a lot of published data about that, and some papers attribute the control of gallinarum and enteritis, which are the most relevant ones, were the most relevant ones in the past. they attribute that the control with use of vaccines. So how much do you think vaccines may have contributed for the control of these two servers, but how much do you think they may contribute for the next servers coming out? Do you think vaccines will be the next tool for biosecurity, or there are other stuff we can use as well?
Vaccines are important, and we can talk about their contribution to controlling enteritidis in a while. But I guess, the main message which I would hammer out to everybody at the beginning, in the middle, and the end of any discussion is that vaccines are only one component of an overall scheme to control salmonella. And biosecurity is far more important than the using vaccines. I know that in hot areas of the world where it's very difficult to control the environment of poultry houses with open-sided housing, biosecurity is difficult. But certainly when it is possible, it is important. Biosecurity is so important. Keeping salmonella out, controlling vermin, controlling rodents, controlling access of birds, controlling contact with other animals, regulating entry of staff, biosecurity and hygiene of staff, regulating entry of people who deal with the electrics and deliver feed, quality of the feed, quality of the water. All these things are absolutely vital. And vaccination on its own will not solve the problem. It will be an important problem, an important component of the overall scheme, but on its own, it will not solve everything. So in the UK, for example, and in the EU, in the late 80s, early 90s, when the UK and EU started to introduce legislation to control salmonella, vaccination was not the major thing that they thought about. They wanted to test and eradicate the enteritidis infection from breeders and then layers, and then also eventually broilers were introduced. And it was only in the later stages that vaccines were introduced. So there was a killed vaccine which was available, and then that was Salinvac, and then a live vaccine from Lohmann, now produced by Elanco. And both of those have their merits. The generally inactivated vaccines are generally less effective because they stimulate antibody, but not very much cell-mediated immunity. But interestingly, I was discussing this with some colleagues the other day, and we came to the conclusion that in any poultry house, birds get infected. Some get infected with a very high dose, some with a medium dose, some with a low dose, and you get a normal distribution of the size of the dose. And it may well be that in the low dose range, inactivated vaccines might still be effective. Live vaccines certainly are going to be more effective, but they can only be a component of the overall scheme to control salmonella. The 9R vaccine is also very effective against Gallinarum and also against enteritidis. But again, controlling Gallinarum cannot be done by vaccination alone. Attention to biosecurity and keeping the salmonella out is absolutely vital.
Okay, yeah. Well, thank you for your answer. It's really interesting what you talked about now. Epidemiology of salmonella is really complex, right? So it makes hard our task to control it, because it's a very adapted bacteria for the environment and for the hosts, especially poultry hosts. Mm-hmm. It's not easy. But in Brazil, we have some changes in regulations now coming out. So some regulations include the control of for some kind of chickens, they consider only Salmonella enteritidis, Typhimurium, Gallinarum, Pullorum, and Typhimurium, monophasic Typhimurium. And for other species, they are trying to consider the control of all servers of Salmonella. How is the surveillance programs in UK, or maybe you can talk about the whole Europe, maybe only about UK? How these surveillance programs consider the Salmonella? Only a few servers, or they survey every kind of server?
They do test for all salmonellas, but they regard Salmonella serotypes which are are most prevalent to be of most significance, I guess. So you're right. In Europe, we don't really have much Gallinarum and Palorum anymore. There used to be Gallinarum in Greece and Southeast Europe in the 1980s, I think, but it's virtually gone. So now really we just talk about Typhimurium enteritidis and Salmonella infantis also. There are occasional isolations from other serovars, but they seem to be less significant. So the Europe concentrates mainly on the major
the most prevalent ones.
Okay. Yeah, absolutely.
Yeah, it's another point hard to talk about control salmonella because we have more than 2,500 servers.
It is, yeah, but if plans are put in place to control Typhimurium enteritidis and Gallinarum and Palorum, then I think you will also find that by biosecurity and vaccination, you will find that other serovars also start to reduce. Because if you're controlling biosecurity, you're controlling the quality of the feed and the water, then the levels of infection of all serovars will start to reduce.
Okay, it's true. So biosecurity is a key for the good control salmonella.
But not just salmonella, it's important for avian influenza, infectious bronchitis, Gumboro disease, everything.
So, Professor Paul, in 2008, the European Union adopted a stringent regulation to reduce salmonella in poultry and eggs, and it worked for years. However, more recently, in 2017, the European Food Safety Authority reported an increasing rate of salmonella detected in poultry layers and even humans. They said that salmonella are not reducing anymore. What do you believe would be causing that return of salmonella? What makes the control of this bacteria no longer an easy task?
My feeling is that, and I've talked to a few people about this, most of the problems are in Eastern Europe, particularly Poland, I think. And my understanding from talking to other colleagues is that people are not keeping to the regulations properly, I think. I can't believe that the enteritidis strain that is being isolated is any different to earlier isolates. Nobody has compared them, but I can't imagine they're that different. And tight regulation of, we were talking about biosecurity, feed quality, attention to monitoring birds and slaughtering when necessary. Maybe this is not done as carefully as it used to be. In the 1990s, enteritis was a huge problem, a massive food poisoning problem, and so people were very, very aware of it. And you could imagine some farmers and companies saying, okay, well, enteritis is not a problem anymore, we don't need to worry about it. And so maybe they're not quite as careful as they ought to be. That's what I think is probably what's happening, I think. I'm sure it is.
There's a lot of new investigations about how should we control salmonella, especially now we're facing some antimicrobial resistance, which also make hard to control it. And one of the fields that we talk about when you want to control some kind of bacteria is the research in bacteriophages. So I know you have a long history researching bacteriophages, and you know a lot about this field. And it's something that comes and goes. I've seen old papers talking about it, and then you see a period of time that nobody's researching bacteriophages anymore. And now I've seen some recent papers talking about bacteriophages again to control, especially the resistant bacteria, which there's no antibiotics that work anymore. So some bacteriophages may do this. What would you say about this research in bacteriophages? What's the potential of this technology? What do you think about it?
It's been an area of my interest for a long time. And the reason was that I mentioned right at the beginning that in the beginning of my career, I worked with William Smith, who was the main person who resurrected the idea of using bacteriophages, because at the time he started work on it in 1980, I think, and at the time everybody said, "Oh, bacteriophage is dead. Forget about it." But he knew a lot about bacteriophage, and he'd used them for typing, phage typing, and he also understood them fairly well. He also had some excellent animal models of infection, particularly the enteritis model in pigs and calves. And he studied that model, but he also studied a septicemia model of E. coli in mice. Now, at the time, well, still now, I guess, that septicemia and sepsis is an important problem. Very often, it's produced by E. coli. One of the main serovars that is associated with septicemia, both in chickens, in calves, and also in humans, are E. coli strains that have a K1 capsule. And it was known in the late 70s, early 80s, that you could identify these strains by the presence of K1 because you could have a phage which was specifically attached to the K1 capsule and killed the bacteria. So it was just a method for identification. But Smith put these ideas together and thought, well, okay, well, maybe we can use the phage to kill the bacteria. And so he isolated a large number of phages, which were specific for K1, containing bacteria. And he tested them in vitro, and he found that some of them were very good, some of them were intermediate, and some of them were no use. So he took the ones which were very good in vitro, and he set up a E. coli septicemia model in mice, and inoculated with the specific phages, and found that it really had a very, very good effect. The other advantage also is that if you use antibiotics, obviously you can get the development of antibiotic resistance. The phage kills the bacteria, and you do get the resistance of the development of phage-resistant mutants. But most of them are resistant because the bacteria have lost the K1 capsule. And that makes them resistant to the phage. But because they've lost the K1 capsule, they're no longer virulent, so they don't produce the disease. So it was a really nice situation.
Wow.
And to some extent, this happened in other models as well, the enteritis model. So I was watching over his shoulder while he was doing this work, and we repeated the K1 septicemia work in chickens and calves, and that was very effective. We've also shown that phages can be effective against salmonella in chickens, and also that phages can be effective at controlling contamination of chicken skin and pig skin also with salmonella. So it does have its place, but I'm having worked on it for a number of years. I'm very cautious about using it.
So when you use phages, you can also have specific species-specific or bacteria-specific action of phages, different from antibiotics that should kill the whole microbiota, something like that. But phages can act only on salmonella.
That's right. They are very specific, but they're also they can sometimes be strain specific. So you can find phages frequently which attach to the O antigen, the LPS of salmonella and E. coli, and so they're just specific for that particular serotype. If you would use phages which attach to capsules, then they're obviously less specific. There are situations where phages can work very well, and generally they are conditions in vivo under which resemble the in vitro conditions under which phages multiply optimally. So you can imagine in the blood, the bacteria mix with the phage very easily, and it's a little bit like the phage growing in a broth culture. Similarly, on enteritis and on skin, it's a little bit like the phage multiplying on the bacteria on an agar plate on a solid surface. So it works very well in that situation. In some situations, it doesn't work so well. So it doesn't work very, very well in the large intestine, where the target bacteria, for example, might be outnumbered by 100 to 1, 1,000 to 1 by other bacteria.
Okay.
It doesn't work terribly well for intracellular bacteria, like tuberculosis, brucellosis, things like that. So there are situations where it doesn't work very well, and you have to be very, very careful. It can work against antibiotic resistant bacteria, and we've done some work recently, which was really quite interesting, because I was thinking about Smith's logic in using phages, that they not only kill the bacteria, but the phage-resistant mutants that develop may be less virulent. And I was thinking about this in relation to antibiotic-resistant bacteria. So a lot of antibiotic resistance is mediated by plasmids, which are transmissible. And they are transmissible because they produce proteinaceous pili, which attach to antibiotic-sensitive bacteria. And these pili are target sites for phages. So we did some preliminary work in vitro, and then also in vivo, and the in vivo work was done at Jabal, in fact, with Professor Berchieri, where we isolated phages which attached to the pili of highly transmissible antibiotic resistance plasmids. And in vitro and also in vivo, we found that the so we had an antibiotic resistant plasmid in a Salmonella enteritidis, and we found that the phages not only reduced colonization by the AMR bacterium, but because it knocked out the resistant bacterium, the small number of bacterial cells in any population which normally don't have the plasmid, the plasmid is lost by 0.1% of the bacteria, these are resistant to the phage. So these started to multiply in preference. And you had replacement of an antibiotic-resistant bacterium by an antibiotic-sensitive bacterium. So it was very interesting. It was very, very interesting. It was very much
modulate the
absolutely modulating
microbiota.
Obviously you have to take these things again very, very carefully, because the plasmid we used was highly transmissible, and so all the bacteria in the culture produced pili and were all sensitive. And many strains of AMR bacteria are plasmid mediated, but the transmissibility is repressed. So although 100% of the bacteria in the culture will have the plasmid, only maybe 10% or less will be producing pili, so they won't all be resistant. So we have to do more work on this to try and modulate it further.
Really interesting point of view, because we always think about bacteriophages only killing the bacteria, but they may also do this kind of inhibit antimicrobial resistance dissemination. Is an interesting approach.
It is, and it's just also made me start to think about the interaction between phages and plasmids. So not only antibiotic resistance, but many other characteristics are mediated by phages. There's a lot of virulence determinants, the hemolysin, K88, K99, the enterotoxin. They're all mediated by transmissible plasmids, so they're all susceptible to these phages. So there must be a very interesting interaction between the phage and the plasmid as well.
Wow, really interesting information. Huh? So, Professor Paul Barrow, as a final message for everyone in the fields working with poultry, the veterinarians or any professional involved in the control of Salmonella, they deal with a lot of pressure, and we know it's a hard pathogen to control. What would be your final considerations to help these people control Salmonella?
I think my main point is what I said earlier, is attention to biosecurity. I think that's the most important thing. I think if you can keep Salmonella out, we don't need to use vaccines or antibiotics. I think, and it's not a magic bullet. It requires a lot of care, a lot of management, and a lot of staff training. But that's really the way that things have to go.
Yeah, and
it's been done in other countries. It's done routinely now in a lot of Europe, and there is information out there that shows people how to do it.
Wow. It's another interesting point you consider there. It's also not only biosecurity, it's not only physical measures, improvements of the physical installations, but also training and education of staff, right? It's an important part of biosecurity plans. When you train, when you give education for the people working in the fields, so they can learn how to deal with that and what they're in contact with. Okay, thank you very much, Professor Paul Barrow. I think this interview has a lot of information for people interested in poultry health, control of Salmonella. This will definitely help a lot of people to understand better what we've been facing, and your experience has contributed so much for this field. I want to make clear that I worked with you for some time in Nottingham. It was a pleasure for me, but more than that, All the works I've been doing with Salmonella were, most of them were based on your previous research or also in the research you've been doing now. And you're definitely a reference for us in Salmonella research. You've contributed a lot for our research here in Brazil. So thank you very much for your participation in this interview.
Well, it was also been very nice to speak to you again, Rafael. And I mean, generally my interactions with colleagues in your group in Brazil have been excellent, and it's always been very nice to host you over in Nottingham and other places also. In fact, it's, you know, I mentioned that I've been working on Salmonella for 39 years, and I think I first had contact with Professor Berchieri in 1988, something like that. So again, we've been worked together for a long time.
It's been a long time. Wow, nice. It's very nice. It's very nice to see that it's still going on, this research, and thank you very much.
Good.